So yes, so I will be talking about representation learning for multimodal image registration today. And most of this work was done during my PhD at Uppsala University. But currently I'm at Karolinska Institute in Stockholm until the end of the year. And then I will be joining the machine learning group at UiT, and I'm really excited about that. So yeah, I'm very happy that I can give this talk today and get to know some of you a little better. So I will first talk about multimodal images in general, and then the registration task of such images, and then how we can use representation learning to actually accomplish registration for such images. So multimodal images in general are images that are captured by different sensors. They give some complementary information about the sample. So you usually are interested in capturing these different modalities and then fusing them together to combine information that they carry. So that can be, for example, CT scans and PET scans or MRI images, and you see that they give very different information about the brain, right? Or up here we have fluorescence microscopy and quantitative phase image microscopy images, or RGB and near infrared. And down here we have some tissue images captured by bright field microscopy and by second harmonic generation images. So the problem is when I capture images with different sensors that very often, in particular with microscopy, they have to be captured in different machines. So they're not integrated in one of the same machines. So when I take my sample here, for example, in the first microscope, and I transfer it into another microscope, the images that I get, they're usually not aligned. So I need to find the transformation that is between those images such that I can actually put them on top of each other and fuse the information that comes from these two sensors. And that task is called multimodal image registration. And it's quite challenging. As you can see with these two images, they're from the very same tissue, and they look very, very different. So the appearance can vary greatly between those images. And the relationship between those pixels is very nonlinear. So very often, the methods that are used for multimodal image registration are based on some local optimization of mutual information, and that is prone to get stuck in local optima very often. So in particular, if there's a large initial transformation, so like a large rotation between those two images, it's quite tricky to actually find the optimal solution without having a lot of restarts of the method. So what we have tried to do is to create some intermediate representations, some intermediate modality between two different modalities that are hard to register, such that we can actually use monomodal registration techniques for those intermediate representations. So here, for example, you have again a second harmonic generation image of a tissue, and here this bright field microscopy image. And what we use is two-stream architecture, two networks in parallel that are combined with the contrastive loss. And they actually can create intermediate representations that we call Comir, for contrastive multimodal image representations, that are similar enough that we can use those monomodal registration methods. So then we can find the transformation, the spatial transformation between those two Comirs, and apply that found transformation to the original images. So, I mean, the machine learning group, probably everyone knows about contrastive learning, but I'll just quickly go into it anyways. So for contrastive learning, you usually have like an anchor that could be a representative of a class, for example, here an apple, or if you don't have any class labels, it will just be one of the samples. And then you have positives to this anchor. So that's either other representatives of the same class or some augmented views of that very object. So maybe like slightly rotated or different colors, etc. And then you have, you are trying to find these abstract embeddings such that the embeddings for the anchor and the positive are close together. And for the negative, for example, for a banana, it would be far away. So we did basically the same, just that for our setting, we want to create 2D representations, and not just embedding vectors, but some image-like embeddings. And I will use anchors and positives for the corresponding patches of the very same tissue. So these two are aligned patches of that very same region in those two modalities. And as a negative, I can choose any patch from the rest of the images from either of the modalities. And these embeddings is basically what we call Comir, and those we can register with monomodal registration techniques. So what we used in this initial study was an influence CLAS to maximize the lower bound for mutual information. And we just randomly crop small patches from our large images during training. And we also sample negatives just randomly throughout the images. And in the critic function, we have found that an MSE, multiple mean squared error, would work best to create those representations for the downstream task of registration. And it was very nice because we could train on very little training data. We do need aligned data, so it is sort of in a supervised manner, but it is requiring quite little in comparison to generative methods in particular. And it was fast to train as well. Code is available on this GitHub page as well. So, of course, we considered using other generative models to translate one modality into the other, and then perform monomodal registration in between, for example, this real bright field image and a GAN-produced fake version of it, based on the other modalities input. So in this case, you would be doing the alignment between this original image and either of these fake images. But what we found, at least for some of the commonly used GANs like Pix2Pix and CycleGAN and JIT, that they could just not produce good enough results for registration, especially with these two modalities that are just differing very, very much between how much content they had in the images as well, since this bright field microscopy image was very dense and very rich with this pink and purple throughout the entire tissue, while the second harmonic generation is quite sparse and has these just black and white structures that correspond to collagen mostly in the tissue. So especially when you're given one of those images, it's quite hard to create a good enough bright field version that will actually allow registration, just sort of impaints some texture that's plausible. And that's of course also limited because we have very little training data for these kind of modalities. And that study was taken further to evaluate different GAN models for these tasks in this paper down here by some of the people in my group at Uppsala University. So they evaluated these Comir approaches versus a lot of different GAN models for other modalities. So they used fluorescence microscopy and quantitative phase imaging, also some medical images, differently weighted MRI images, and also other benchmark data sets of these two microscopy types. So down here, you can see some more examples of these bright field images that are being produced by GANs based on the second harmonic generation input here. And yeah, the structures that it captures are maybe plausible as tissue, but it's just not good enough to perform registration. One thing that we found quite important when we produce these representations are equivariant properties to them, since when I have a multimodal image pair as an input during one iteration, it will be treated as an independent sample then in another iteration where this image pair is rotated. So there's no connection that it has to learn between rotated versions of it. But since my downstream task is registration, I have to expect that my test images will be at least rigidly misaligned. So what I need is that these representations, that a rotated Comir or the Comir of a rotated input will be actually the same as as the rotated Comir of that input. So I'll just quickly go into rotational equivariance versus invariance. So for classification tasks, for example, if that's my downstream task, I usually want some invariant mapping as a network. So when my input, this dog here, rotates, I still want my label to be the same no matter in which rotation I give this input. And that's invariance, rotational invariance in that case. But for 2D representations that I try to learn with my network, so for example, in segmentation where my label is a 2D mask, I want the output to rotate in the same way as my input. And that's rotational equivariance. And that's also what we need in this case of 2D representations. So what it really means is that if T is my spatial rotation, my spatial transformation, and F is my network, I want to have this commutative diagram that in case of equivariance. While in invariance, no matter if I apply one of those spatial transformations, I want it to map to the same thing. And rotational equivariance can be either learned or it can be achieved by network design. So learned equivariance is usually done by data augmentation. So that can be useful in segmentation tasks when I just show differently rotated versions of my input together with the rotated label mask that I want to learn. For equivariance by network design, it's a bit different. Then I try to create each layer of the network to be an equivariant function. And then as a composition of equivariant functions, my entire network will be an equivariant function. And that I can either realize for convolutional layers by lifting convolution, which is generalizing Euclidean convolutions to group convolution, or by steerable convolutions. In the case of Comirs, we have explored a different version of learned equivariance that's sort of applicable to contrastive learning. So if, for example, my two differently colored ducks are my input for my two networks that will actually produce Comirs, what we have done is that I will give one of the inputs at 0 degrees, but my second input, I will actually have a 90-degree rotation to it. Then I train my two networks and get my feature maps that are also like 90-degree rotated between each other. And then I just rotate the feature map back by the same transformation that was applied to the input before I actually evaluate the loss. And that in that way, I managed to have my networks commute with the rotations quite easily without any additional hyperparameters that I need to take care of. And it's probably a less strong equivariance than if I have it by network design, but it is quite easy to implement and works very well when you have contrastive loss. Initially, we have only considered 90-degree rotations for the simple fact that we didn't want to deal with the interpolation of the feature maps. And we were actually really happy to see that that together with the data augmentation that we had in the training, it would actually lead to equivariant representations for any degree, basically. So here, what you can see is a stabilized view of of our input images. So here in this case, we have an RGB image of a house, or like a bunch of houses and some forest here, and the near infrared image of that. And if I don't have any equivariant constraints on the network to learn these representations, you can see these two Comirs being produced from these two input images. And then just sort of one of the input images is rotated, but then the Comir is rotated back, so you can have this stabilized view to compare them. And you can see that they are learning these shadows that goes along with how the error changes, the transformation changes between the input images. And that's definitely not what you want. So when we have this loss that commutes with the rotation, we get these images here. So they're quite similar throughout the rotations. And something is not working quite well with the video, unfortunately. But if we don't have this equivariant constraint, we get this blue line here, which shows the correlation between the two Comirs when one of the input images is rotated. So we get like a very nice correlation at 0 degree and like a decent one at 180 degree. But in between, we get quite a difference between those Comirs if we don't have these constraints. While in this orange line, you can see that we get almost a fully nice correlation throughout all the degrees, even though we're just requiring these multiples of 90 degrees to be really aligned. That's the C4 group. So that was very nice for us to see. And here's just some results from that paper that introduced Comirs. On this data set of bright field microscopy and second harmonic generation images, these purple and black and white ones. The red line here was the state-of-the-art for the registration that was in place up to that point. And what you would ideally want to see is like a curve that goes all the way up to the left corner here, because it is it shows how many images we were able to register given a pixel error that's given on the y-axis, on the x-axis, sorry. So ideally you would want 0 pixel error and 100% of them being registered. That's definitely not the case. But what one has to consider with these images is that they are actually not ever captured in the same microscope. So the ground truth is already manually annotated. So we can't really expect a 0 pixel error. So what we also had is a small user study with, now I don't remember, like 10 annotators, I think, that annotated a subset of our test set. And that's given with these dotted lines. So that's the pixel error that humans were able to achieve with these images. So that kind of means that like a pixel error of 20 pixels is quite reasonable to expect with this data set. And then in green and orange, that's the line that we get with Comirs, using either alpha AMD, which is an intensity-based registration method for monomodal images, or SIFT, a feature-based registration approach. So we were able to get fairly good results on this sort of data, even though clearly there's like a lot of the improvements can be done on this registration anyways. So one of the following up studies that we have done with this approach was to see if we can also use this for the formal registration, because in the initial study we have only considered rigid registration. So that is one study also done in the group that I was in, where they extended this learned equivariance to more general transformations. So on the one hand, rotations of all degrees, and also fine and deformable transformations. And then use these representations to for deformable registration tasks, together with this Inspire framework for that is also a monomodal deformable registration method. That's quite relevant for microscopy images, because very often when you have multimodal images, you can't use the same sample in all microscopes, because they might need a different sample preparation. So what you have to do is you take consecutive slices, so like a slice of the tissue that's very close by, and then there's some underlying deformations that you have to consider. And the same for medical images. So very similar to our initial setup is that one of the input images gets transformed, for example, and here is an affine transformation, while the other one is kept normal. So you have to follow either the red path or the blue path. Then the networks are trained, and then the same transformation is applied to the feature map before the contrastive loss is applied. And they also had some pretty good results on that study. One more thing that we've been working on now is to take Comir into 3D, which is, there's like nothing hindering it. It's just an implementation, like we hadn't gotten to it initially. So we have one master student working on evaluating, learning these representations in full 3D and comparing it to learning the Comirs slice-wise in 2D and then do the reconstruction before registration. And we had initially thought that learning in full 3D would likely be work much better. But so here you have an example of two of these Comirs for two MRI modalities. And they have more detail, but we have quite some issues with this upsampling, as you can see here. While here we have two versions of learning them slice-wise. And we are still evaluating the results that we have from that study. But one thing that we could see is that when we learn these representations slice-wise, we just have a lot more training samples, like a lot more pairs of anchors and positives than when we are working in full 3D and I can only crop so many cubes out of my brains for training data. But at the same time, when I work with the full 3D cubes, I do have more information per sample. So there's this aspect that we try to explore a bit more in the study. We have also used these Comirs for cross-modal image retrieval. So when you have an entire data set of, for example, this bright field images, and you're given one query in the other modality, and you try to find the match within that database. This would be that one in this case. And what we had done here is also to like encode the entire database into Comirs and my query patch, and then use a bag of words where I extract some surf features in that case that I cluster into global image descriptors, and then I can match them with cosine similarities, retrieve my matches, and then we done some re-ranking of the top matches on top of that. And that worked pretty well as well for these Comirs. But when we were studying these 3D Comirs and saw these artifacts that I just showed from the upsampling, we wondered why we were actually using a UNet at all. We had just kind of started doing that initially, but realized we should actually try some fully convolutional networks, like a ResNet, for example, and see how that would work instead of using these UNets. And that worked pretty well. So we started working on the one hand, changing the architecture for these representations, but also adjust the loss to encourage properties that are particularly relevant for registration. So on the one hand, using the ResNet architecture, it allows us quite easily to use group convolutions. So we get these rotational equivariance properties by network design. And we realized that with the ResNet, our training was actually even more stable than with the Comirs. It was working already pretty well with Comirs, but we needed, for example, gradient clipping to make the gradients not explode. That's something that we don't observe with these new representations. And we were able to train on very little computational resources, so basically just a laptop with the GPU. No more computational resources for this now. And we observed that we can go down with the training data even more. So with the Comirs, it varied for modalities that were pretty similar, like this RGB and near-infrared images of aerial images. We could train on just one image, but these microscopy images that I showed, this bright field and second harmonic generation images that are very different, we trained on, I think, 35 images, which is still like very little, I think, in comparison to what you need for the GANs. But here we could go down even further with the training images And this is like an ongoing study, so we're still looking at like how little training data we can actually do with. So they differ quite a bit from the Comirs. We call them emirs, so intermodal image representations. And what we do is that it's not a contrastive loss anymore. So we only have an MSE-based loss between the two positives. So if you have here my input modalities, my modality A and my B, these are my two networks producing these emirs here that you can see, and I have a loss between those positive ones to get the intensities of these features as similar as possible. Then I have a mutual information loss between the input of modality A and the emir of modality B. That's directly maximizing the mutual information across those modalities, and the same vice versa here. Then I also have a gradient aligning loss that goes across the modalities. That's this loss term here. That one is based on normalized gradient fields, which is also a measure, or like a similarity measure, or a technique that's very often used for multimodal image registration. And then to ensure that the features are actually retaining like a lot of detail and not just sort of default into kind of like a binary dark gray and light gray version, we also have a distribution matching term that is acting on the entire batch. So there I am basically taking the histogram of all the intensity of the intensities of all the pixels throughout the entire batch, and I want to spread them to a target distribution. So in our initial studies now, it's basically a uniform distribution to spread the pixels out, the features out a bit, to retain more detail. But we hope to work some more on maybe matching the distribution of the original modalities, but that's still ongoing work. But so far we can see that we get a lot more detailed representations than we had with the Comirs. So here, for example, you have the original images of this data set. It's an RGB and a near-infrared image. And these two images would be these two representations are these emirs that we generate with our new approach, while these two would be the Comirs that we had. And one thing I think that you can see with the Comirs is that it kind of favors these blobby structures. Like it kind of creates round structures if it can, which I think is from this rotational equivariance, since like something round will definitely be rotational equivariant, so it's a safe bet. But with these emirs, we can we want to keep some of the high frequencies which we get from this gradient aligning term, and yeah, by spreading the features uniformly, more or less, we also get some more details in the structures. And here's another example of the initial Comirs from some brain images versus some of these new representations that we've been working on right now on these emirs. So you see there's more details being preserved. Yeah, so in summary, we showed that we can use representation learning to generate 2D embeddings of multimodal images that can serve as some sort of intermediate modality that allows us to transform this multimodal task to a monomodal task that is just generally easier, and we have more registration techniques that we can use for them. And these approaches require very little aligned training data, especially compared to like fully generative methods like GANs or diffusion models. But of course, we are also interested in trying to get to a place where it's like fully unsupervised if possible. And we were able to use intensity and feature-based registration methods, both for a rigid and deformable registration, that we could apply for these representations. So yeah, thank you for your attention, and the code is available to basically all the studies that I mentioned now from my former group, and I'm happy to answer any questions.